and welcome to this episode of Gen Live entitled Beyond Baby KJ, Next Steps in Manufacturing Genome Editing Therapies. I'm Juliana Lemire, Deputy Editor-in-Chief at Gen, and I'll be your host today. So it was announced in May that a baby known as Baby KJ, born with a genetic disorder, was treated with a gene editing therapy tailored to his rare mutation. This was a first in the genome editing world. The milestone made headlines and marked a huge win for the genome editing field, offering new promise for the future of genomic medicine. But there's another layer to this story that is of fundamental importance to the success of the therapy, but did not really get as much airtime, and that is how the treatment was manufactured. Developing a single personalized therapy required a coordinated sprint from teams at IDT, Aldevron, Danaher, Acuitas Therapeutics, and more, working under intense pressure to produce the gene editing payload and its delivery system. Their hard work paid off. The teams did something never done before, and the baby's doing well. Nobelist Jennifer Doudna said that this work opens up a new era for treating previously untreatable genetic diseases. And that's what we're here to discuss today, because making just one treatment like this was a huge effort involving many teams. In turn, delivering treatments like this to more patients is an enormous challenge. So on this Gen Live, we take a close look at the work behind KJ's successful treatment, but perhaps more importantly, we'll explore how this can be done on a larger scale in the future. We will discuss the challenges that lie ahead, including scale, safety, and cost, and how some companies are thinking about solving them. To do that, we welcome an amazing panel of experts. First, we welcome Sandy Atensman, Vice President and General Manager of the Gene Writing and Editing Business Unit at IDT. Sandy has spent the last decade within Danaher's life sciences and diagnostics business at IDT for over 3 years now. Before joining IDT, Sandy served as VP Global Marketing and Product Management at Phenomics. Previously, Sandy was with Beckman Coulter. Thank you so much for joining us today, Sandy.
Great, thanks for having me.
We also welcome Mark Wetzel, who joined Aldevron in 2021 as transition leader following Danaher's acquisition of the company. After that, Mark became Vice President and General Manager of mRNA Services at Aldevron. And before joining Aldevron, Mark held positions at both Leica and SciEx. Welcome to the show, Mark. Thanks for coming on.
Happy to be here. Thank you.
Lastly, we welcome Dr. Sadek Qasim, a scientist and executive with extensive experience in the biotechnology industry with a specific focus on cell and gene therapy bioprocessing and translational research. Currently, he serves as CSO and CTO at Danaher with a focus on genomic medicines. Previously, he held positions at Vor Bio, which works on autoimmune disease, Kite Pharma, and Mustang Bio, who both work on CAR T-cell therapy. And earlier in his career, he worked in Novartis' cell and gene therapies unit and with research teams at the NCI and the UPenn Gene Therapy Program and Johnson & Johnson's Immunology Discovery Group. With Sadek, it's more a question of what hasn't he done than what has he done. It's a pleasure to have you join us today, Sadek. Thanks for coming.
Thank you, Juliana. Thanks for the kind introduction.
And before we jump in, I want to remind our audience that we would like to answer your questions too. So please put any questions that you have for Sandy, Mark, or Sadek into the Q&A section of the platform, and we'll get to them as soon as we can. So as I said, it's been 2 months since the Baby KJ story was announced at the ASGCT meeting in New Orleans. So we've had a couple of months to digest the news and all the publicity that surrounded it. But for those who may not be familiar with this story, I think it would be a good idea to start with a recap. So Sadek, if you don't mind, I'm going to ask you to give an overview of the story. And I know you've done this several times, but unlike stints on CNN and others, you have more than 30 seconds. So please take it away.
Great. Well, thanks for that introduction, Juliana, and thanks for inviting us today to share the story of Baby KJ. I'll start a couple of years before Baby KJ. This all started with a collaboration that we announced with the Innovative Genomics Institute that was founded by Dr. Jennifer Doudna, who won the Nobel Prize for describing CRISPR gene editing back in 2020. And so our partnership, Danaher, including Aldevron, IDT, Cytiva, which, by the way, for those that aren't familiar, Danaher is a global life science and diagnostics company. And so really our main mission is to enable drug developers, academics, translational investigators to translate their ideas into the clinic and into breakthrough therapies and diagnostics. And so recognizing the tremendous potential of CRISPR gene editing, but the limited access to gene editing in general, we partnered with the Innovative Genomics Institute around a partnership, what we call a beacon, the Danaher IGI CRISPR beacon for CRISPR cures. The aim was really to platform gene editing and to make it more accessible to the thousands of genetic disorders that can be addressable by gene editing, but are limited by logistics related to manufacturing and just overall infrastructure. So that was the genesis of our partnership with the IGI. Now, the director of the IGI Danaher Beacon for CRISPR Cures, Dr. Fyodor Urnov, is also part of an NIH-funded consortium called the Somatic Cell Gene Editing Consortium, which is tackling the concept of platforming gene editing, but really from the basic research and preclinical side. So as part of that consortium, there are members across the United States, different academic centers, two of whom include Dr. Rebecca Arns-Nicholas, who's at the Children's Hospital of Philadelphia, and Dr. Kiran Musunuru, who's at the University of Pennsylvania and is a co-founder of the company Verve Therapeutics. So Kiran and Becca had been funded by the NIH and the Somatic Cell Gene Editing Consortium to platform gene editing for inborn errors of metabolism. So these are errors or diseases that affect the liver and impact a newborn's ability to metabolize, basic metabolic pathways. So they had developed a preclinical path towards going from initial genetic diagnosis to preclinical screening to developing a research-grade drug product for inborn errors of metabolism. They've been doing this for the last 3 years thanks to NIH funding. And then when baby KJ was born a year ago, he was born in August of 2024, they quickly recognized that he was born with sort of the symptoms of something called a urea cycle disorder. And the symptoms typically include jaundice, the inability to metabolize protein, and just really stunted growth. So what they did is they rapidly deployed targeted genome sequencing, were able to identify a mutation in an enzyme that affects the urea cycle disorder. And typically the symptoms of this include elevated levels of ammonia in circulation. And so the particular mutation that KJ had was fatal. The 50% probability of death within a year after birth, and then after that, the mortality rate increases significantly. So they were racing against the clock. They had worked out the entire workflow for developing a custom gene-edited drug product, but the missing piece was the manufacturing piece. And so via our partnership with Dr. Urnov and the Innovative Genomics Institute, Kiran and Becca reached out to Fyodor. Fyodor reached out to us and said, look, we have an opportunity here, but it's going to require certain capabilities and it's going to require us to move fast. Less than a year. Ideally, manufacture this drug product in less than 6 months. Danaher, IDT, Aldevron, can you meet this challenge? So quickly I brought in Sandy, who heads up the guide RNA business at IDT, and she'll tell you where the story went from there. And Mark Wetzel, who heads up the mRNA drug product business within Aldevron. Quickly we started to brainstorm, whiteboard, see whether it's logistically feasible. Can we achieve this unprecedented timeline and deliver something that could be potentially, alleviate KJ's symptoms? And that was last August, September roughly. And, from there, you see the headlines, but I think the details of how things were done, really Sandy and Mark take all the credit there, because it's really their teams who were burning the midnight oil, trying to figure out logistically how to make this happen between Coralville, Iowa, which is where IDT is based, and Fargo, North Dakota, which is where Aldevron is based. and a whole slew of academic and industry partners, the team was able to pull off in less than 6 months something that had never taken place. And so that's really the story up until the time of manufacturing.
Okay, perfect. Thanks so much for that overview. And I think exactly right. I think we let Sandy take it from here. So Sandy, when Sadik called you and said, this is what we need. I mean, actually, so from your team at IDT, what did you need to produce, and how did you go about, getting all of that together to get it ready on time?
Yeah, absolutely. So from IDT's perspective, as I think probably most people know IDT, we're an oligo company, right? So we make the guide RNA, and we've been doing that for research use for a long time now. And we opened our cGMP manufacturing about 2 years ago to be able to make, therapeutic quality guide. And so I think it was Sadik, if I recall, we were in person a meeting. I think it was actually in August where he had first mentioned it to me. To do this, that, hey, this might be coming. And I want to say it was maybe a few weeks later in September where I think it was the first call. I believe, Sadik, it was you, it was me, Mark, you were there. Like, we had a couple of us there where we started to talk about it. And so from my end, so what our team did is we did the guide RNA. And my first question was, well, what's the sequence and how hard is this? Because we knew with this timeline, I think overall, like start to finish, we've estimated in a best case scenario, you do this sort of thing when you think of both the guide, what Mark will talk about with the mRNA and with the LNP and the final drug product, that takes at least 18 months. And we're talking, we want to dose by February and we're in September, right? So do the math there. We think of what would normally anyone we're working with who wants a guide, you're wanting to do a pilot, you're wanting to see, can we make the sequence? Are there challenges? Is there anything funny in the sequence that might make it a little bit more complex to synthesize? And so we had to pretty quickly figure out, is that doable? What can we do? How do we streamline this? And then the other piece we also worked on them with is, from an IDT perspective, is we offer a lot of work around off-target analysis as well. And so we were doing, that's the nomination and the confirmation to understand, is this guide going to do any off-target edits that you don't want? And so the team also wanted to work on that with CHOP and UPenn on this guide, but also, as you'll hear, I think from Mark, we'll talk about in a second, this was a novel-based editor. So we hadn't ever customized any of our services to do that. So we were having to work very closely with them and with Aldevron and everyone on that part as well. So it just, as I said, a lot to do, and from our perspective, it was for us getting the guide as quickly as possible, both a first batch for toxicology and then an actual final batch for the drug product, so that our drug substance, so we could get that to Aldevron as quickly as possible. Maybe that's a good point for me to pass it to Mark for him to talk about what Aldevron did on this part for the other part to actually make the drug product.
Perfect. Yeah. Thanks, Sandy. Mark.
Yeah, thanks, Sandy. So yes, we got the call in September, and we mobilized the team. And while the science is fairly straightforward here, despite the novel base editor, it was the timeline that was really daunting. Our optimized processes get us to a end-to-end solution, LNP RNA, typically in 10 to 12 months, but this was a 6-month target that we were after, which was, again, pretty daunting. So by the way, this was all partially enabled because of our relationship with Acuitas. We mentioned Acuitas in the beginning, but they're a highly collaborative partner. They had a clinically relevant LNP with their experience there. That also is a way that we really optimized and accelerated the timeline, the pathway via the FDA was laid based on that. So we quickly engaged Acuitas in the discussion, and it became a sit-down workshop where we got the people in the room and we mapped out the critical path. Something that Danaher, I think, is good at is executing and focusing on results and keeping the end in mind versus the 1,000 potholes, as I like to say, and reasons why we can't hit this timeline. So with that mindset that the team had in this room, we really mapped out, what can we do given the timeline? This is what the target is. What is possible versus what can't we do? And it was a very productive session. You know, it was, hey, go get this person and see what they're doing, pull them from their desk, let's get them in the room and talk through a potential solution. And, you know, that's how we did it. But this became a highly complicated sort of program management exercise, and with a real focus on execution for our teams.
And Mark, can you just in case people aren't familiar with what Aldevron does, I mean, can you just tell us, you know, you got the guide RNA from IDT, and then you got the LNPs from Acuitas, and then, I mean, it's a CDMO that's going to put this all together, right, and turn it into something. And then I imagine that was the final product that got shipped to Pennsylvania. Is that right?
Yes, thank you. Aldevron is a company well established in the nucleic acid space, well known for plasmid. We're a CDMO. Upon the acquisition of Aldevron by Danaher, my role was to pull in the technologies available throughout the platform to build out our end-to-end capabilities for LNP and RNA, which uniquely positioned us here for this opportunity, right? We have all of these capabilities more or less under one roof, which allows us to be really efficient and minimize the tech transfer, for example. But Aldevron is very experienced in RNA. We've been making RNA since 2017. We've produced 150 GMP RNA batches at pretty sizable scale. So again, as I mentioned, the science for us was fairly straightforward. It was really an execution problem.
And I mean, I imagine, just like on a personal perspective for a second, Mark, I mean, you sent this to Philadelphia, I imagine, on dry ice. Is that how it goes?
Yeah, I mean, the cold chain, you know, especially coming out of the COVID and the COVID era, the cold chain supply chain logistics is very robust. So yeah, we were able to get that to CHOP pretty quickly and without risk.
Okay. I was wondering how it felt to kind of send that box out.
Yeah, this is the thing, Juliana, right? I mean, there was no loss for you know, purpose with the Aldevron and the IDT and the Acuitas team. You know, this noble purpose that we were rallying around. So while we were running a business and finding a way to dedicate the time and resources, you know, to find a way to get this done, required that people come in nights and weekends and work really hard and be creative. And it was not, I will say, not that hard to get people to rally around the purpose here, which is a key part of why we were successful in dedication of the teams.
Yeah, terrific. And so, actually, just before we move on to kind of lessons learned from all of this work that was done, I want to make sure we're bringing in all the groups. I mean, definitely Acuitas is important to mention, but I think other groups maybe were involved, like Jackson or others. I just want to make sure that we give credit where credit is due.
Yeah, no, it was really a constellation of collaborators between academia and industry. You mentioned Acuitas, which helped partner with us on the LNP front, as Mark mentioned. And then in terms of the academic design of the guide and the base editor, because this was a custom editor, as Sandy mentioned, that was a collaboration with David Liu, who's at the Broad Institute, and Ben Kleinstever, who adapted and optimized the base editor specifically for Baby KJ's mutation. And then Jackson Labs, Dr. Kat Lutz, she developed a custom mouse model that recapitulated the genetic mutation on a mouse background to enable some of the initial safety screening. And then, you know, we had a few other groups, but that was really the major sort of groups that were involved here, in addition to the Children's Hospital of Philadelphia and UPenn, obviously.
Yeah, just a huge collaborative effort, I'm sure.
That's
so
right.
Having gone through it, you know, what are some of the biggest lessons that each of you learned in the last year of doing this work? Do you think about that? I'm trying to think, like, moving our discussion at some point into kind of going into the future and doing this more and more. You know, is there something that you learned that you feel like just is a critical component of what will be brought into future work in this area?
Yeah, maybe I'll start with my thoughts on that one, and it was kind of building off something Mark talked about earlier, which is he talked about how we had to look at the schedule and look at the planning, and I think one of the big things that I think for us was how do we take really thoughtfully calculated risks and risk benefit for this, especially given the patient. So obviously there was a lot of discussions that our joint teams for IDTL, Devron, and then CHOP-UPenn were doing with regulators on what is appropriate for a patient who doesn't have time to necessarily wait for a year or 2 years, right? And then there are also things that we made decisions. So one example is, again, IDT has a separate our manufacturing facility for our guide RNAs in Corvallis, Iowa. We had to ship that to Fargo, North Dakota. And we shipped all of our products under quarantine without having finished QC. Now, we did all the right things we had to do, but those are things you normally would never do, right? In this sort of business, you would never send something without having like done all of the QC and having that completely finished, but we had to save every day. And I think those were the sorts of decisions that the teams were making. We parallel processed a lot of things. We started some things earlier when normally you would wait for contracts to be 100% finalized or certain data to come in. And you take that risk, which normally you wouldn't take as part of a business, but we did because we had to look into every single way to save time. And I think that was something that I take away from this, of getting people to think differently and to realize, when are you willing to take a little bit of a risk to that, because having those extra days, those extra weeks count so much.
That's super interesting, Sandy. I mean, how do you think about, let's say that, in the future you're creating a standard workflow for this type of work. I mean, can you be cutting those corners in a systemic way, or no? How are you going to build that?
Yeah, I mean, I think it's a great question, and I think I want to be careful when I say cutting corners, because it's like there's like cutting corners and there's parallel processing, right? It's like taking the risk. Like, we never took a risk on a patient cutting corners. I make sure that, but you're taking a risk on cost, and I do think that's important, because normally you don't do it because you don't want, say, send it to Aldebaran and they go do a lot of work with it and like, whoops, sorry, we had an error, you have to redo it. I think those are the things we're going to have to look at, right? So the ideal is, how do you make that faster? How do you make all of those steps faster? How do you do that? And there might be cases where you have to be willing to say, all right, we're going to have to take that sort of cost risk to take this time out, or it shows us where we need to take time out in the future. But I think those are the sorts of things that normally when you do this, you're minimizing all of your risk, and that risk includes cost risk across the benefit and then across the process, and having to understand where do you have to take some of these steps, and if you're really trying to get to these time frames.
Mm-hmm. Yeah, and I apologize, cutting costs was my words, not yours, so I'll strike that from the record. Mark, what about yourself and kind of lessons learned having gone through this,
A couple things come to mind. For us, we never viewed this right out of the gate as a commercial opportunity, obviously, and we got support top to bottom from the organization to make that happen. While we did receive some funding, as Sadek mentioned, some sort of token funding. It was never viewed as a commercial opportunity. And the other really constraint that we had that we can't overstate is the ability for the legal teams of all these organizations to come together to really align on how we were going our working relationship, right? And that took some time, but really, again, at the center of all of those discussions was the patient, right? Which is what made it happen. But just to add a little bit to what Sandy said, we learned a lot about what's possible operationally as we went through this. And in addition to taking risk and, optimizing our handoffs throughout the process, we also learned a lot about how to make things more efficient by pre-planning, queuing up materials, doing some practicing on equipment in advance, just so that we were ready when the material came and that we could produce efficiently. So those are the kinds of steps that we took, and those are the lessons that we can take away and reapply going forward. So again, we learned a lot about what's possible here within our own operation.
Sadek, do you have something to add? Yeah.
Yeah, I mean, from my perspective, I mean, I think just the remarkable collaboration across the ecosystem between industry, academia, and the regulators as well. FDA was engaged from the very beginning in terms of the pre-IND all the way to the final IND approval, which is really remarkable. Then the other part is the NIH funding was really critical to that foundational build that Kieran, Becca, and the academics were working on, Theodore, the IGI. And then I think the other part relates to, you know, just the convergence of technologies that are available to actually enable even treating someone like a KJ with this particular mutation. So it really required, I mean, when you think about it, when you zoom out, what we have here is two Nobel Prize winning technologies, mRNA, which won the Nobel Prize in 2024, gene editing, which won the Nobel Prize in 2020, coming together in the formation of this drug product. But then you also have these innovations like base editors, which can target a single mutation. You have LNP, which enables targeted delivery to the liver. And you have sequencing technology and next generation sequencing to enable on target. So it really required all of these technologies to come together, be stitched together in such a way that enabled a coherent workflow. And I think that's sort of the remarkable aspect as well. Like, I don't think this could have taken place in any other ecosystem outside of the United States, nor in any other time. So it's really the convergence of technologies, luck, and willing partners across the ecosystem that enabled it. And now I think the responsibility for us, or the mandate, is how can we recreate what was lucky the first time, but how can we actually double down and make it sustainable, so that it's not luck per se, but it's actually a standard process that can be used to enable treatments for more baby KJs.
Well, that was exactly my next question, Sadek. So I'll throw that right back at you. I mean, when you think about this, what does need to be done in order to bring this to more patients? What would you say are the biggest things in your mind right now? And then I think we should walk through some of the challenges separately, like, scaling up, safety, things like that.
Well, I'll start off with one aspect, and then I'll hand it over to Sandy
Sure.
and Mark from the operational. I think from a regulatory perspective, the unique thing about gene editing as a platform is that you can theoretically, because as we mentioned, there are three components to the gene edited drug product. There's the LNP, which is the formulation that you're using to deliver the drug substance, the payload, the therapeutic payload. And then the payload itself is made up of two components: the mRNA that encodes the base editor or the gene editor, and the guide RNA, which is like the GPS system that will track the payload specifically to the affected region in the chromosome. So theoretically, when we talk about a platform, you can retain two of those elements, the LNP and the base editor, and simply swap out the guide RNA, which will be very much patient and mutation specific. And so in this way, what you could do is you can scale the process of drug development. You don't have to go through a traditional drug development path where you have to revalidate all three components each time for each new patient, which could take years and millions of dollars. Here, once you've validated two of the three components, the LNP and the base editor, you can simply swap out potentially the guide RNA, which will be very much patient specific. And what this does is it gives you scale from a regulatory perspective, because now, we have over 7,500 genetic disorders, most of which are Mendelian disorders, meaning driven by a single mutation. And right now, there are no economics incentives or regulatory incentives to develop drug products for all 7,500 genetic disorders, which impact over 300 million patients worldwide. However, when you platform, now you have a regulatory framework that enables you to quickly develop drugs almost on demand, like we did with Baby KJ. But I think with each new turn of the learning loop, you can potentially compress that timeline. So although KJ's drug has been called the fastest drug ever developed, less than 6 months is what a lot of observers have viewed. I think there's a world of possibility where we can even compress it to below 6 months. I mean, maybe even a month or 2 months or 3 months. theoretically, once you've platformed the drug product. So I think that's one opportunity for scaling, is really implementing a regulatory framework that enables you to scale, not just for one genetic disorder, but for potentially all monogenic Mendelian disorders. So I think that's one opportunity to scale from a regulatory perspective.
I have questions on that, but if Sandy and Mark want to add to what you just said, I think that would be great.
Yeah, maybe I could add to that, since, one of the things that Sada called out there is you can have this platform and you're keeping the LNP the same, you're maybe keeping the mRNA editor the same, you still need to have a separate guide every single time. So that's definitely not escaped me as with our team who makes that guide every single time for every single patient, right? And I think one of the things, I think there's some benefits on that. I think one benefit of being IDT and the way we approach this is we have built our business off of coming up with oligos, synthesizing them very quickly in large quantities, right? I'm actually sitting in our facility where we make literally hundreds of thousands of oligos a day and hundreds of thousands of oligos overall, and then literally thousands of guides a day, right? But those are not the quality you need in a therapy, right? But we can apply a lot of those learnings to how do we get this faster and how do we get in the right quantities, which are very different than if you're thinking from like a CDMO size. Of that, how do we get that faster? The big piece that we have to talk about, and as we work with regulators, is terminology we kind of consider phase appropriate, right? People are used to getting this as full cGMP, and for those who know that production, that's a single suite, single product. There's a lot there, both on a cost basis, but also on a time basis that takes to do that, right? A lot in terms of getting that done. So we have to work with regulators on, is there something we can make that, what is the right risk benefit analysis for the patient if the time is so critical? And how can we think about working with them? And we're really talking about this through our partnership with the IGI on how do we work with regulators, and how do we work on this platform with the kind of coming up with phase appropriate material that is the correct risk benefit for these patients. That's going to be really important. I think it's really important for the whole product, but it's really important for the guide RNA, where you're thinking that's the piece you're going to have to maybe like change every single time for each one of these patients.
Mm-hmm.
Yeah, for me, I mean, all of that is relevant for Aldevron and the RNA. I would say the opportunities fall into sort of four buckets for me in the way that I think about it. One is applying the lessons learned operationally that we gain from the KJ story, and our continuous improvement engine. How do we make our operation more efficient is a focus that we're working on every day. So that's one. The second is we, like many, if not all CDMOs in this space, are not set up to operate at this small scale, right? We're manufacturing volumes of RNA, much larger scales. I mean, we overproduced here because that's what we had to do. So there's a lot that we can do in terms of innovation here, technology platforms, continuous RNA manufacturing, for example. But also we talk a lot about AI and the application of AI, right? We've talked about it in the way that it can benefit the front office, but there are places that we can apply this in construct optimization and also in our operation, importantly in this case, where, can we auto create batch records? Can we detect deviations and diagnose deviations with using AI? There are just some opportunities there for us to focus on technology. So that's two. The third, of course, is what we've talked about in the regulatory pathway and the platform and the phase appropriate, the acceptance around those concepts, and really, expanding our use there. And then, of course, the economics is the fourth bucket, the reimbursements, the way that the system views these curative therapies versus treatments. That all has to change. That's a stepwise change that needs to happen. So those four things is what I would say we look forward to. But I think we're hopeful in all of those areas that there'll be progress.
Yeah. Okay. Terrific. I have so many questions, and we have so many questions coming in from the audience, which I want to get to too. I want to go back to one thing quickly, Sadek, that you were saying, and the platform. So many people are talking about platforms right now because it seems like the way to go. But I mean, I imagine for some time, like the next baby KJ will be probably, I would imagine, another N of 1. Correct me if you think I'm wrong. And I know a lot of that regulatory work to get the N of 1s was really pioneered by folks in the ASO space, Stan Crook and the people who are making not genome editing treatments necessarily, but N of 1 rare disease treatments. So how do you go about kind of like going from N of 1 regulation, which is so distinct from other drug regulation, to a more platform-based regulation? I mean, you'd have to go through a clinical trial, I imagine, right?
Yeah, I mean, I think it's a great question. I mean, we are standing on the shoulders of many pioneers like Stan Crook with his M. Loram Foundation, like Julia Vitarello and Tim Yu, who've also demonstrated N of 1 in the ASO space. So I think there are learnings from those particular spaces. I think, one thing to consider here is that with gene editing, it's regulated by a different branch of the FDA, CBER, which, I think the risk-benefit profile of a biologic versus a small molecule drug is slightly different. There may be some harmonization, but I think that could be something that this field of gene editing can leverage. And there's been quite a bit of discussion, starting with the scientific exchange that was held back in November by the FDA, and then the subsequent roundtable that was held by Secretary Kennedy and his group there at HHS about leveraging the learnings from Baby KJ to really enable a regulatory framework. So I think there are, to be sure, many unanswered questions about what does a platform look like, what's the minimum clinical evidence required to enable platforming across multiple diseases. But I think what I see with the Baby KJ story and just the momentum in the field relative to other previous efforts is there does seem to be momentum in terms of a path to figuring it out. Mm-hmm. And I think people see that if you have these potentially curative therapies, there are many things that need to be figured out, and we'll just prioritize those things. There's the scientific and technical, there's the reimbursement that Mark mentioned, there's the operational. So I think we're not there yet, but I think we with this Baby KJ story, I think we've collectively this partnership has provided the field with the possibility of what this could look like, and now it's really reducing to practice and standardizing and getting the buy-in from other players that are working in developing treatments in this space.
Okay, terrific. I want to switch a little bit to some audience questions. So somebody has a question about off-target analysis. I mean, I guess I'll add, what was done to check that. And also they ask, is there any way to scale that as well and make it faster and more efficient?
Yeah, I can take that. So the teams at IDT did off-target analysis, and we did both nomination and confirmation. So those were using UncoverSeq, which we actually published around the same time as this all came out, and we started offering as full service, which is IDT's version improved upon GuideSeq, if people are familiar with that for the nomination. And then we did confirmation with RampSeq, which again, IDT sells as kit, but we also do as a service, but as that, and we supported, these were supporting the filings that they did with the agencies for approval, right? So these were really important. We supported that. I think the thing about how do we make it faster, one of the things the team did that was, going back to the burning the midnight oil we talked about earlier, is because it was a new base editor, we had to actually customize it for that. But what you would imagine is we have this set up for different editors, and then we are working on it all the time to make, how do we parallel process it. It really doesn't take that long. It's compared to everything else in the time it takes to do it, once you have it customized for the type of editor and the type of work you're doing. So that's something we're working on and trying to, for this one, we'd be able to then do it again faster than we did the first time, but it's always something we have our bioinformatics teams working on to try and improve. But it's a great question. I think it's a really important area that we want to talk through is how do we make sure this is safe, right? Because we want to make sure that we are editing the right thing, that we are minimizing this, and so making these tools as accessible as possible is something we're working on.
Okay, great. Another great question kind of about how this may change the industry is this an audience question from Chris. Do you think that there will be a trend toward in-housing of LNP engineering formulations for gene editing companies? Or do you think that there will be a trend towards consortia partnerships between nucleic acid companies and delivery-focused companies like LNP companies?
Yeah, I mean, I think there's no monopoly on innovation, so it's going to continue to be a combination of both in-house and external partnerships. But I think each one of them has their trade-offs, pros and cons. And I think part of the reason that in this case we worked with Acuitas, or the CHOP and Penn worked with Acuitas, is the prior experience in other gene editing trials. And I think this is part of the lessons learned as well. Kieran had founded a company called Verve Therapeutics, which was working on the largest killer of individuals, worldwide cardiovascular disease.
Right.
And he extracted those learnings from Verve to enable an N of one. And I think the lesson learned there is that, although we're developing, in the KJ story was a drug product custom for one individual, a lot of the lessons were actually leveraged from a larger indication, cardiovascular disease. And I think there is this two-way communication in terms of things that are done for large indications will benefit N of one, and vice versa. Every patient that we treat from an N of one perspective will also help the broader field in terms of accelerating development for larger indications. So I think there is that two-way street that, the shared learnings are going to, I think, benefit the field in general.
Yeah, actually, and that feeds into great comment. Another question from the audience. And Mark, you touched on this a little bit earlier, talking about needing how you overproduced and you kind of did it this time. But I imagine long term, maybe that's not the plan. But anyway, they ask, well, first of all, they say fantastic seminar. So thanks to all of you. And then they say, from a technical perspective, are there challenges to scaling down? For example, right now, your manufacturing scales are meant for N of many. Will there be a need for new technologies needed to be fit for scale on demand and a few therapies? Even mundane things like cleaning in between guide RNA manufacturing runs in order to de-risk cross-contamination. It feels like new processes are going to need to be implemented.
Yeah, there's really no question there. I touched on it a little bit earlier, but this innovation in the manufacturing space has to happen because we're just not set up to produce these small scales repeatedly. Things like continuous RNA manufacturing that eliminate some of the steps you just described in the process is one opportunity. AI, as I mentioned, is another potentially, but we've got to find ways to scale down and meet the needs here for sure. So technology and innovation is definitely part of the future pathway here.
Okay, terrific. And there's about half of the questions are about the cost. So I will summarize them all and say, how is this going to be paid for in the future? How are we going to bring the cost down, and then who's going to pay for it?
But I think the first part is how do you bring the cost down, right? I mean, I think there are questions on how much did it cost. I mean, I think Mark said it really well earlier. Like, we didn't see this as a commercial opportunity, so we didn't go track all the costs of exactly how much this would all be. And again, when I share stuff, we had R&D teams doing stuff that you would only have to do the first time, right? So when you think of the costs and how you do it, I think what we have to do is work on, as Mark was saying, is on the manufacturing. How do we get the right manufacturing scale to produce at the right level? If we're overproducing, if we're building a small amount of science in a space that's meant to make 100 times X more, that's obviously not going to work, right? It's like that math that isn't going to work. So we have to on the manufacturing. And then I think going back to what we were talking about earlier with the phase appropriate as well too, is we need to work with regulators on how do you think of the amount of testing? What do you need to do? What is cGMP? What is appropriate when you're working on an N of one or an N of few versus you're working on a bulk that's going to 100,000 people, right? Like, I mean, they're very different risk calculations you need to have, and we need to work with them on it, because if you can get that rules and we can agree on what that looks like and have some of these novel manufacturing processes then approved, then you'll be able to drive the cost down significantly for that.
And just to build on what Sandy is saying, I mean, I think it's obviously, I think people appreciate that with any new technology, the first time you do it, it's going to be really costly, but then with each new iteration, costs will go down. I mean, sequencing is a really great example. Just in the last 20 years, price of whole genome sequencing has dropped 100,000 fold, and the infrastructure that's been built to support sequencing is very different today than it was 20 years ago. Rockets, for example, when we first sent a man to the moon, that was cost prohibitive, but now we have reusable rockets that are being sent into space on a regular basis. And I think that reusability and just that scaling down and making things fit for purpose are really going to bring the cost down significantly. And I think the advantage with something like mRNA and LNP is that it's a potentially completely synthetic process. So you could start off with a cell-free DNA template, synthesize that potentially, synthesize mRNA. I mean, it's an IVT reaction, LNP. These are chemical components for lipid components. So I think, as you go towards more chemical-based processes or synthetic processes, part of the costs are also going to come down as well. And I think the other component in terms of cost, it's not just the production, but it's also the analytical characterization of the drug product. And so automating those aspects and doing the phase appropriate or risk benefit appropriate type of analytical characterization of the drug product will also bring down costs.
Yeah, thanks, Sadek, for calling out the analytics. My team would probably be kicking me if they were in the room here for not calling that out. But analytics improvements, technology improvements, and innovation is another really key area that's going to help us optimize the timeline here. But it's really back to those 4 buckets. How do we get the cost down? It's improvements in all of those spaces. It's our own internal efficiencies, it's technology innovation, it's the regulatory pathway, and with a focus on platform and phase appropriate, and lastly the reimbursement mechanism.
Well, and to Sandy, to your point earlier, I guess we don't really know how much it costs. I mean, it's my understanding that this work was an in-kind donation, correct?
Yeah, we got, I think Sadek said it earlier, or Mark said, we got a token amount, small amount for it, but yes, it was mostly in kind.
Okay, so I mean, I guess the cost that number will be determined at some point.
Well, because you also have to remember, Juliana, that as I mentioned, Karen and Becca, Dr. Arns Nicholas and Dr. Mosinuru had been working for a couple of years on developing the preclinical platform, NIH funding through the Somatic Cell Gene Editing Consortium.
Right.
And then there are other players in the ecosystem, like, for example, the National Institute of Standards and Technologies, NIST, has been working on this consortium for gene editing for a few years to enable standardization in the way we characterize these things. I think there's a lot of ecosystem costs that enabled something like a Baby KJ that we benefited from. But then in parallel, there were the internal investments that Aldevron and IDT and Acuitas had made that we were able to leverage and tap into. So I think, the cost, even if we did have the specific number, it would be just arbitrary for what's possible. I mean, this is what was possible 6 months ago or a year ago. But even in that time, from last year to today, I think the cost would be significantly lower than, last year, just because of operational efficiencies that Mark and Sandy refer to, among other ways of working. So I think this is just going to continue to decline over time.
Okay, terrific. Now I just lost a question I was going to ask. That's okay. So I have a question, which is, let's say in some number of years now, I won't ask you to put a number on that number of years, but maybe 5 or 10, that these therapies are gaining momentum and, everything that we're talking about today is coming to fruition. So what new challenges do you think might be opened up as these medicines do become more available?
I mean, something that comes to mind for me is the access, the equality of access.
Okay.
You know, the availability of these therapeutics across all demographics, something I think about a lot, right? We can't have this be a treatment or approach for the privileged only, right? So that's one thing that comes to mind.
Yeah, and building on that, I think, sometimes we take for granted the learning curve for physicians as well to implement these technologies. So I'll give you an example, CAR T therapies, and this is well known, even though these are potentially curative, and I worked on a couple of these during my time at Novartis and Kite at the National Cancer Institute. Despite them being launched a few years ago, they're still only accessed by about 20% of eligible patients in the United States. And it's a mixture of, lack of access to the community hospitals, but also physicians not being familiar with these technologies. So we do need to match the technology and scientific innovation with clinical, coursework and for the next generation of physicians. And, one thing that Dr. Musunuru mentioned is that things like the baby KJ treatment are going to, lead to the rise of new disciplines like interventional genetics, where we train physicians how to sequence and intervene at the genetic level, like we do with other disciplines like interventional cardiology. So I think, the science and technology will enable drug production and manufacturing, but I think there's also that need to train future physicians and clinicians about the possibility of what can be done with genetic medicines. And I think there's always that gap in terms of the reality of what's available versus the standard of medicine or the practice of medicine that's implemented in community hospitals and even academic centers.
Yeah, Sadek, I just want to put an extra point on that last piece that you brought up with, like, without Becca and Kieran and their understanding of this doesn't happen, right? And what I mean by that is, like, you don't have to have someone who's designing the drug, like they designed, like they knew what to do, right? Like they have an understanding. That is a rarity, right? That you're gonna have those type of physician scientists who are there working. So it's like, what we need to do is, to what Sadek's saying, to get this not just out of like the most elite like academic medical centers and doctors understand, but how do you get it that you can have the tools and everything in place that it's almost like a design your own, CRISPR editor for this type of genetic disease. I don't know, maybe you're looking 5-10 years in the future, you need that sort of tool to be able to do a lot of this, right? Or some way to have that work.
Yeah. Well, and that's why, I mean, maybe in a sense, the enormous amount of publicity that this story did get, hopefully brought it to many, many, many people, and then, physicians and non-physicians alike, but might, open up some doors there as well. So that's why science communication is so important, I guess. So another audience question, beyond the liver, what are the most promising next frontier organs for in vivo gene editing within the next 3 to 5 years? And I guess that brings up the question of, are we going to go beyond LNPs here for delivery mechanisms? This story, I know the context is just LNPs.
Yeah, there's a lot of good work being done on AAV as a delivery vehicle as well for gene editing for things like CNS, indications. But I think with non-viral, lung, kidney, and even, people are working on local ophthalmic indications, CNS. But I think in the short term, lung and liver will probably be the lowest hanging fruit, and then they're followed thereafter by kidney and local administrations like ophtha.
Okay. And I guess I will, since we're kind of almost at the end of the hour, I guess I will just ask for a little bit of a prediction from all of your perspectives. Over the next couple of years, what do you see happening over the next couple of years? Where do you see this going? And how quickly might this move to the platform that we were talking about? Does anyone want to take that?
It's a big.
End on a tough one.
Yeah. Well, yeah, maybe I could start things off. I mean, I think you mentioned, Juliana, the coverage that Baby KJ has brought to the field and the possibility of what can be done with today's technologies. And I think, this coverage has gone global. So there are many geographies, outside of the United States, Europe, Middle East, South America, Asia, that are very interested in figuring out how they can enable this in their respective ecosystems. And I think, what that will do is it'll enable us to access innovation globally, bring the best minds to work on really solving some of the challenges that we talked about. But I think deploying this in other geographies, what it does as well is it, forces us as a field to figure out a way to enable access, and really work for the future, both from the diagnostics and also from the intervention perspective. So I think, what I'm most optimistic about is the potential globalization of these types of technologies and just the global interest that, this has triggered from different health systems in different countries worldwide. So I think this will be, hopefully, you will see more individuals treated on a global level, and it really enabled us to be accessible by many, not just in the United States.
Yeah, I'd probably add to that. I mean, everything Sadek said, but I think I've been really excited of where we've been. I think I mentioned earlier our partnership with the IGI, the Innovative Genomics Institute, recently announced the Chan Zuckerberg Initiative is investing with them as well in order, how do we do more of this work. I've been really excited with just the work that our teams are doing at IDT and Aldebaran with the IGI in order to start pushing this forward. And I think that's what I mean, it's kind of seeing where I think of where that started 2 years ago and what we're talking about now. Like, I think if you had told us 2 years ago that's where we would be here when that was kicked off. I don't know, Sadek, you were very involved in that. I'm pretty sure we'd be very excited, like, that type of progress. I'm just excited where we're going to be able to go from here now that, like, we have gotten more of this global attention and more, hopefully, investment in others in these initiatives to make it move forward.
Yeah, not much to add for me. I'm hopeful despite the macro environment we're in right now. I do believe the science will prevail, like many of us, and there are 300 million people waiting. So I'm hopeful.
Well, I cannot think of a better note to end on than that one, Mark. I am also hopeful that science will prevail. So with that, that will bring us to the end of this session of Gen Live. I want to again thank all of you, Sandy, Mark, and Sadek, for this terrific discussion. And I'm glad that we gave manufacturing some really good airtime. So we'll do it again.
Thank you for the opportunity. We really appreciate it.
Thank you.
Thanks to all of our audience for tuning in today, and make sure you join us on our next Gen Live. We'll be discussing organoids on that one. So I'm Juliana Lemure, and I'll see you next month on Gen Live.